The vasculature serves as a conduit for the transport of red blood cells, carrying oxygen around the body. Due to their biconcave shape, red blood cells flow down the central line of a blood vessel. As a parasite matures within an infected red blood cell, the cell loses its biconcave shape and becomes rigid. The deformation causes the infected cell to be pushed out to the walls of the blood vessels by a process called margination. Performing adhesion assays under conditions of fluid flow allows researchers to investigate how a parasite binds under conditions closer to those found in vivo. There are many options available to researchers who wish to study this interaction. For example, capillary tubes, parallel plate chambers, self-fabricated slides, and commercially available slides. Using the following protocol will allow you to assess the ability of malaria-infected red blood cells to bind to either endothelial cells such as primary HBMEC or the immortalized HCMEC D3. The following protocol uses microslides from Antibodies. While slides are being coated with collagen, start to prepare the cells for seeding. Preparing a blood smear. To check on the health of the parasites and to count how many there are, prepare a thin smear. Add a few microliters of blood to a slide. Using another glass slide, hold it at a 45-degree angle and gently pull it toward you. Capillary action makes the blood flow along the edge of the slide, and when you push it along the glass surface, a monolayer of cells is formed. Fix the cells by dipping in methanol. Parasites can be visualized by staining with Giemsa. Then allow to air dry. To read the slide, add 1 to 2 drops of immersion oil onto the slide and read with a light microscope. Parasites appear as purple-blue bodies inside red blood cells. Parasite MIA is quantified by counting the number of infected red blood cells and dividing that by the total number of red blood cells multiplied by 100. Connecting slides to the pump. Once the parasite suspension has been prepared, it is time to connect the slide to the pump. First, attach the 3-way valve to the 20 ml syringe. Then place the 20 ml syringe into the pump and secure in place. Connect a smaller syringe to the 3-way valve. Then connect the plastic tubing to the remaining port on the valve. Finally, connect the tubing to one of the reservoirs on the channel slide. Protocol for flow assay. When running the assay, set the flow rate to the desired micrometer per minute rate to generate 1 dyne per centimeter squared. Allow the parasite suspension to flow over the cells for 5 minutes to allow binding. Switch to wash buffer and flow over for 3 to 5 minutes to remove non-bound parasites. Count the number of infected red blood cells bound per millimeter squared. Repeat these steps for each condition being tested. Example results. Here are some examples of the types of data that can be generated using this assay. It is possible to record the level of binding over a range of different shear stresses on either endothelial cells or recombinant proteins. You can add antibodies targeting surface receptors such as ICAM1 or EPCR to determine their contribution to adhesion. It is also possible to use slides coated with recombinant proteins, for example ICAM1. Here the data shows the effect of different antibodies targeting PFEMP1 domains and their ability to abrogate adhesion to ICAM1 under fluid flow. Flow assays allow for not only measuring levels of adhesion, but to measure other interactions such as rolling. Here you can measure the velocity of a bound parasite as it rolls across cells. Another phenotype that can be measured under flow is rosetting, where an infected red blood cell binds non-infected and forms rosettes. This can be measured and quantified under conditions of shear stress. This video is an accompaniment to Chapter 42 and can be found online. Many thanks to our funders for supporting this research.